Alright folks, we're picking up with question 38 on the biology air practice test. So question 38 is asking about it's modeling allele frequencies. That means basically how often an allele, a version of a gene, shows up in a population. So each graph in this question is looking at, basically they're showing the allele in 3 different trial runs. It's a computer simulation, and the red, the blue, and the green are just 3 different trial runs. And they're looking at how the gene can vary over 50 generations, how much these genes might change in an example population. And so what you're looking at is we'll start down here, one of the graphs, we see some changing in values for each of the 3, but it seems to be very consistent. The graphs are all very close to this 50% frequency. We see another, the top one here, which has some variation. It's a little bit more dramatic, but again, it stays pretty close. We don't have any extreme cases here. There's some big swings, but nothing too crazy. And then number 2 here has some really big swings where the green line goes up to 100%, where one allele takes over. The blue line goes down to 0%, where the allele they were testing disappeared. The red one jumps way up and then way down and then back up again, so there's some dramatic variations in that one. And the only real point of what they're trying to get to is the amount of variation in the size of these swings is related to population size. And that makes this question really easy. The bigger the population, the less impact some variation will have, like some swings in the number of individuals with a certain gene. So if the population is big, it's going to have the smallest swings. So 3,000 individuals, our biggest population, is going to be the graph with the smallest amount of variation happening. 300 then goes to the next smallest, and then 30 individuals is the one where we might see some really dramatic swings in some of their alleles. So that's this part. The second part says the graph that shows the greatest impact on genetic variation would be this one. This is the one where the genes can vary the most. So again, this is 3 different trial runs, and in each one we have some dramatic variations happening. So this is graph number 2, and that goes here. So that's that answer. Next up, we're looking at DNA replication. How does DNA copy itself? The word that is most important here is semi-conservative. DNA replication is semi-conservative. The phrase, or the term semi-conservative, means, in this case, that half of the DNA is conserved or kept, and the other half is new. So this is blue indicates one of our original strands. Every new strand of DNA that's built is built in yellow. So blue is our original strands, yellow would be our new strands. So there are multiple answers here depending on how you divide these up, but mostly the first step here is there's only one good answer, I guess. The first step involves this one and this one again, because again, semi-conservative. We see half of this original DNA. One strand goes this way, the other strand goes this way, and each strand gets a new strand as a partner. So the blue strand is the original, the blue strand here is the original, and then our 2 yellow strands are the new ones. Okay, so we get a second round of replication where this DNA strand is now going to divide again, but this time it's already got a yellow strand. So let's say the yellow strand goes this way, the blue strand goes this way, and we're going to make new strands again. So since we already had a yellow strand and we're going to make a yellow new strand, it's going to be both yellow. On the other side, we've already got a blue strand down here, and we're going to add a new yellow strand. So it's going to look like that. Now again, I could switch these 2, and they would still be true, because again, I would just change the direction that these 2 went. These 2 have to be both half and half. These 2 are going to be one half and half, and one that's all yellow. It doesn't matter which one is which. You could switch those 2. But that's the correct response here, again, because DNA replication is semi-conservative. This DNA strand will split into 2, and then a new strand forms with it. And then this will split into 2, and a new yellow strand forms with it. So that's how we fill that one in. Alright, next up we have an electrophoresis. The question is asking you about trying to find out if this insect-resistant gene, which is we see here in the resistant sample, to see if that gene is making its way into the population of a particular crop. And so we look at these different samples from the crops. This is DNA sample 1, 2, and 3. And we're looking for if any of these samples seem to match the resistant sample. And for electrophoresis, again, the key is if we're looking at the same gene with the same allele, we should see a matching set of bands. So DNA sample number 2 has a matching set of bands as our resistant one. It's probably got the same gene, and that's how we pick that one. The other two seem different. They probably do not have this gene. Alright, next up, this is called a dichotomous key. A dichotomous key, in this case it's for birds' feet, is a way to classify things based on what characteristics they have. It gives you a guide on how to sort them out. So we have a foot of a bird, and we're going to use that to decide what species this bird is. So you start with step 1, and it's pretty straightforward. 2 toes or 4 toes. So you select which one that is. In this case, 1, 2, 3, 4 toes. So 4 toes says go to step 2. So we go to step 2. 2 toes forward and 2 toes back. 3 toes forward and 1 toe back. 3 toes forward, 1 back. That's the correct one. So we're going to, again, follow what it says. Go to 3. So we go to 3. Are the toes webbed or not? They are not webbed. Go to 4. We go to 4. Toes in a curled position. The toes are curled. That tells us that it is this Vermivora Chrysoptera. If they are flat, it means go to 5, and we look and see what their toes are like. So, but in this case, the toes curl here, so I'm going to say this is a Vermivora Chrysoptera. That's the bird where this foot is attached. So, Vermivora Chrysoptera is letter C. That's my answer. Alright, this question is fairly straightforward. We have a cladogram. All I want to know is what information about an organism, or about the organisms in this cladogram, will best help scientists determine the evolutionary relationships among them. There are lots of nice ways to do it. Their anatomy, their habitat types, maybe. Their reproductive strategies, maybe. But the best one, by far, is to look at their DNA sequences. The more similar the DNA, the more closely they should be related to each other. So that's the best answer here. So DNA sequences, letter A. Next up, another cladogram. This is found in a biology textbook. The branching diagram most likely used to represent the theory that suggests what? What theory are we talking about that fits this diagram and what it represents? And what we're looking at here is really this. Letter B. All these species that are involved, all these little branch endings, represent a species that's either here now, or a species that was here like these guys were and then died earlier in the past. See, time going forward. These guys are here now. These guys were here and went extinct along with this guy too. These ones went extinct because they're not here anymore. These ones are here now, so they're still around. What we're seeing is a common ancestor. That's why these guys join together as we work our way back in time. This is a common ancestor to everybody on this cladogram. Alright, these are common ancestors. This is a common ancestor to these guys and these guys. Everywhere we see these guys join together, that's a common ancestor. And so over time, change occurs where these common ancestors split into different groups, split into different smaller subgroups, and that's where these different curves, these different directions come from. So this is letter B. That's the best answer. So next question. Which statement describes the role of adenosine triphosphate, that's ATP, in a cell? ATP, remember, is like the energy currency. Its job is to store and release energy within a cell. Remember, if a cell needs energy, typically it's going to use ATP as its source. And that's why cellular respiration, for example, its entire job of that process is to make ATP. And then that ATP is what's used to do all kinds of other things within the cell. So that's letter B. Next up, we're talking about evolutionary relationships and how to tell if two things are closely related or not. I listed several of them here, so you could just kind of read over them. I'm going to go over them real quickly. The most reliable options I put first. Comparisons of DNA. Again, the more similar the groups are, the more similarity you're going to see in their DNA sequence. So like if they're 98% the same, that's very similar. If they're 80% the same, that's more distant. Number 2, same thing with proteins. You could do the same thing with a protein chain because they're made up of these amino acid sequences. It's the same way as DNA. If they're very similar, that means they're closely related. If they're very different, that means they're not. Number 3, you can compare anatomy. Similar groups tend to have similar underlying structures, things like their bones. We talked about in class the forelimbs, the front limbs of different groups of like mammals. So like a human forelimb and a whale and a bat and a cat. Even though they look different on the outside, they've got the same structures underneath, like their bones. And then you can use that for living things. You can also use that for things that are extinct, because you can look up you can find their fossils and do the same kind of analysis. Last, we did talk about embryological development and how things that are closely related tend to develop in similar ways. So the more closely related you are, the more similar you see during embryological development, early development. And that happens even when the adult forms are very different. Alright, we'll stop here and start the next video with the next question.